into the last, actually, week of our semester. We have final exams, comprehensive lecture final exams next week. So I made 3 PowerPoints, just selecting a few slides from each of our lecture unit, and I posted those. And my hope was to just really, really quickly, very quickly, just go over some of the slides and just mention a few topics that you might see on the comprehensive lecture final. And realize, folks, this is nowhere near complete, right? Because it is a comprehensive final exam. So students who did well on the final exam in previous semesters said they would use their lecture study guides to prepare. Okay. And one thing really quick, you guys, today have been in about 4 hours of Zoom meetings. And one thing that came up is for fall of 2020, we will still, the campus will be closed. We'll still be sheltering in place because of the pandemic coronavirus. And for those of you that are going to be enrolling in classes for fall, you'll see that some are labeled as asynchronous. That means that all of the lectures will be recorded so that you can view those lectures at any time that you want. And then that's asynchronous. I hope I got the vocabulary right here. And then synchronous is that you will be meeting for lecture and or labs online at the scheduled time, as in the class schedule. Now, most of us, I think, in the biology department, we're going for what's called, if I can remember this correctly, synchronous hybrid. And that means that some of the activities will occur during the scheduled lecture and lab times, and then others will be recorded. So those would be asynchronous. So for example, if you have maybe folks that you know of, for my fall 2020 Bio 440 sections, I had it classified as synchronous hybrid. And that means that most of the lectures and labs will be pre-recorded so folks can watch them when it's convenient for them, as long as they're staying up. Um, um, but just as you guys have experienced online, our lecture exams, our lab exams are during the scheduled class time. And likewise, any lab quizzes would occur during the scheduled lab time. So I just wanted to give you guys kind of a heads up there. We thought it might be a little bit confusing for folks trying to register for fall. So I hope that might help a little bit. Okay, guys, so again, we're going to go through these slides so fast just to kind of give us an idea, maybe refresh our memories. And I think maybe people were most concerned about the units we covered for the face-to-face lecture exam 1, and that was Unit 1, kind of the intro to microbiology, Unit 2, biochemistry, and Unit 3, cells. So we'll just go through these slides, you guys, and again, this won't be comprehensive, right? But hopefully just kind of it will help jog our memory. Hopefully, we hope. Okay, all right, you guys. So, so in the introduction, our Unit 1 introduction to microbiology, you'll recall that we did a little bit of history. We had you do some history homework. So I just listed here, folks. Um, um, these are important figures from microbiology history. They'd be good to know. So Leeuwenhoek, who was the first to report the existence of microbes. Hook, he was the first one to use the term cell for the basic unit of all living organisms. Edward Jenner was the first one to use cowpox virus as a smallpox vaccine. Semmelweis was the Austrian physician who recommended that doctors and medical students wash their hands before examining their patients. Pasteur was brilliant. Oh my gosh, he did so many things. Disproved the theory of spontaneous generation. He helped prove that microbes are everywhere. He did important foundation work for the germ theory of disease. He developed the first rabies vaccine. He helped save the French wine industry, developed pasteurization. Those are just a few. And then a contemporary of his, Robert Koch, a German physician who succeeded in proving the germ theory of disease by developing 4 postulates, 4 prerequisites to show that a specific microbe causes a specific infectious disease. Okay. Florence Nightingale, who used statistics to show that hygiene, good diet could help decrease death deaths in the military, right? So kind of the heroine of nursing. Joseph Lister, who read of Semmelweis's work and Pasteur's work, he developed aseptic techniques during surgery. He had carbolic acid sprayed over the patient, the incision site on a patient during surgery. He had bandages soaked in carbolic acid. He had people um, rinsing their hands in carbolic acid, right? Um, thus, thus greatly reducing the number of hospital-acquired infections. John Snow, the father of epidemiology, using mapping, he determined that the source of a cholera outbreak in London came from water from the Broad Street pump. So he had the handle of the Broad Street pump removed. Didn't know that cholera was caused by a bacterium Vibrio cholera, but he proved by doing mapping you could determine at least a source, a reservoir of a pathogen. Darwin, right? Darwin, famous for describing biological evolution as a product of natural selection. Paul Ehrlich, the first to develop the concept of magic bullets, chemicals that would selectively harm or kill a pathogen without harming the patient. Um, Alexander Fleming, who discovered the first antibiotic, penicillin, produced by the fungus, the mold Penicillium, and Fleming also discovered lysozyme. Watson and Crick, who described the structure of DNA, and then Carl Woese, who developed the three-domain classification system using ribosomal RNA sequencing. This is a great link, you guys, down here. It has a timeline and then a brief description of some of these significant folks in microbiology history. And then, folks, in that Unit 1, really important was our idea of classification and our ideas of biological evolution and natural selection. So these are a couple of themes, right, in biology, the unity and diversity of life. So as biologists, we explain the unity of all living organisms as a result of us all descending from a common ancestor about 3.6 or 3.8 billion years ago, the first cell. The first cellular organisms evolved, and we're all descendants, right? So we share many traits with all cellular organisms. And then the diversity of life, as time progressed, we have this great branching, diversification of cellular organisms, and that's a result. One of the processes was natural selection, where as organisms moved into different environments, the environment would select which variants in a population were best adapted, right? And those variants would survive longer and have more offspring. And so gradually over time, the genetic makeup of a population of organisms would change, and that's the definition of biological evolution. And the beauty of natural selection is that over time a population of organisms become better adapted to survive in a specific environment. And here's just kind of a repeat of what we just went through, right? And then, folks, classification, both in lab and lecture, you'll remember classification was really important. So here we see in this tree of cellular organisms the three domains, domain bacteria, domain archaea. And we want to remember that all prokaryotes are found in domain bacteria and archaea. And then the third domain to which we belong is domain eukarya, right? And here are, these would be examples of kingdoms. So kingdom Animalia, to which we belong, kingdom fungi, kingdom plantae, right? And you'll recall, folks, that we said that what used to be known as kingdom Protista is no longer used. In our course, we called protists, we used a lowercase p. We describe them as mostly unicellular eukaryotes that were neither animals nor plants nor fungi. And another thing, folks, we just want to remember in general that the more closely related a pathogen is to the host, the harder it is to find magic bullets that will selectively kill that pathogen without harming the human host. And then, folks, again, this is kind of a dichotomous key for classifying all microbes, all agents, all organisms. So remember, classification will be really important, you guys. Right, so do you remember acellular? We have our prions that are disease-causing prions that consist of misfolded proteins only, no nucleic acid. And then also acellular are viruses, who consists of protein and nucleic acid, either DNA or RNA, but not both. And remember, you guys, viruses are obligate intracellular parasites or pathogens, right? And the reason we classified prions and viruses as being acellular, just by answering one question, could the agent synthesize a cell membrane? If the answer is no, they're acellular right off the bat. But then, folks, if we come back and ask, does the agent or organism synthesize a cell membrane? And if the answer is yes, then they're cellular, right? And we divide cellular organisms into two basic groups based on the absence or presence of a nucleus. So if it's a cell, and these are almost all unicellular organism, and they lack a nucleus, they're prokaryotes. And remember, there's two domain archaea and domain bacteria, right? If a cellular organism has a nucleus, then they are eukaryotes. They belong to domain eukarya. And the four groups we looked at was the protists with a lowercase p, right? Eukaryotic microbes that are neither fungi nor plants nor animals. And then we looked at kingdom fungi, kingdom plantae, kingdom animalia. We talked quite a bit about the endosymbiotic theory, and the endosymbiotic theory states that eukaryotic mitochondria and chloroplasts evolved from primitive endosymbiotic bacteria, the mitochondria evolving from primitive endosymbiotic aerobic respiring bacteria, and chloroplasts evolving from primitive endosymbiotic cyanobacteria. And consequently, folks, since mitochondria and chloroplasts evolved from bacteria, we would predict that we would see 70S-like ribosomes in mitochondria and chloroplasts. That's at least one piece of evidence supporting the endosymbiotic theory. And then, folks, in Unit 2, on our final exam, there won't be a lot of specific biochemistry questions, but since all our microbes are built from atoms, right? So it's like all of microbiology basically is biochemistry. It is important that you know kind of the basic ideas. It's like if we're going to build our prions, our viruses, our cells, right? We need to know the basics of biochemistry. So we know water is really important, right? Water is a polar molecule. It has two polar covalent bonds. And it can form a maximum of 4 hydrogen bonds here, right? Which leads to many of the emergent properties of water. And something that will be really important, you guys, is knowing, and probably you're dreaming about hydrophilic and hydrophobic, right? So it is really important to know the difference between hydrophilic and hydrophobic. So hydrophilic water is attracted to hydrophilic substances because they're either polar molecules or they have a complete charge, they're ionized. Hydrophobic are going to be our nonpolar substances such as lipids. And then protein structure, you guys, will be really important. So we need to know the 4 levels of protein structure. So the primary level of structure is the specific amino acid sequence of the protein, stabilized by peptide bonds between amino acid residues, right? And that primary structure, the specific amino acid sequence, will determine all higher levels of structure, and thus will determine the functional conformation of the protein. So secondary level is when we have bending into alpha helices, beta pleated sheets, right? And then we're going to have further folding into complex 3-dimensional shapes, the tertiary structure, stabilized by R group water interactions. For some proteins, this is a functional conformation. But in some proteins, we want to remember there's 2 or more polypeptide chains. And thus they will exhibit what's called tertiary structure, and that's the arrangement of the polypeptide subunits. And again, that's going to be stabilized by R group, and R group water interactions. I just threw this in here, you guys, because as part of the immunology section, we did talk about the structure of IgG. So IgG is an example of a protein with quaternary level of structure because we have 4 polypeptide chains, 1, 2, 3, 4. And if the polypeptide chains aren't in the correct quaternary structure, the antibody can't function, right? And just throwing this in here, you guys, remember these arms are called the FAB fragments. They contain the antigen binding site. That's what AB means, antigen binding site. So there's one there and one there. And then the third fragment is the FC fragment, and this is the part of the antibody to which complement will bind to be activated. And it's also the part of the antibody to which phagocytic cells have receptors to bind to this FC end. Protein denaturation, usually within cells, we say it's irreversible. If we denature, we unfold our proteins. Then, even if we remove the denaturing agent, the denatured proteins, when they try to fold up again, they can form inappropriate R group, R group, R group water interactions. So you end up with permanently unfolded proteins which can't function, and that's usually lethal for the cell. And then, folks, we talked about diseases caused by abnormal protein folding, and the one, of course, we really focused on were diseases caused by misfolded prions, right? So the good normal cellular prion here, the misfolded prion here, they have the same amino acid sequence. Isn't that wild? The same primary structure. But you'll remember that when the prion misfolds, we have changes in secondary, tertiary, and quaternary structure, right? So we get these misfolded prions. They stack together, they form long filaments, and somehow those filaments in the brain, for example, trigger the neurons to die. Right? So we end up with a family of diseases known as the TSEs, transmissible spongiform encephalopathies, right? Literally spreadable, wholly brain disease, 100% fatal. There's no treatment. There's no prevention as far as we know, unless it's not eating tissues from animals that have prion diseases, right? And the other bad thing, you guys, you'll remember that we mentioned that our immune system can't identify the misfolded prions as being foreign. So there's no immune response, there's no antibodies, so there's no easy blood test, right, to detect antibodies against a prion to see if somebody's or an animal is incubating the disease. The misfolded prions are some of the most resistant biological structures known. So remember, if you're eating beef from a cow that had the BSE, the mad cow prion, cooking the beef really well won't inactivate the prions. Normal autoclaving won't inactivate the prions. Most of our chemical antiseptics and disinfectants won't destroy the prions. So, for example, if you were doing surgery on a patient with a known prion disease, probably the best thing to do would be to incinerate the instruments so they're never used again, right? And you will recall that, and I think it's the third, final exam PowerPoint. I did put more slides in on prion diseases. So some of the human prion diseases, such as Kuru, spread through mortuary cannibalism, the Creutzfeldt-Jacobs disease, which kind of happens spontaneously, and then the heartbreak, the variant Creutzfeldt-Jacobs disease, which we believe is caused when people consume beef from cattle suffering with BSE or mad cow disease, and then the cattle prion binds to the human prions and triggers the human prions to misfold. So that's quite a heartbreak. So again, 100% fatal, and there's no treatment. So then we're still in biochemistry, folks, but again, so many of these are just foundation concepts for later units. So we then move to nucleic acids, right, DNA and RNA. And I think we started the unit saying, well, if the amino acid sequence of a protein is so important to its functional conformation, what determines the amino acid sequence of a protein? And of course, the answer was the DNA sequence of the protein's gene. So if we have a mutation, if we change the DNA base or nucleotide sequence, that could change the amino acid sequence of the mutant protein. And if we change the amino acid sequence of the mutant protein, that can change its shape. And it might lose its shape, it might lose total function. But sometimes, what's so cool, you guys, is sometimes when there's a mutation and you alter the amino acid sequence of a mutant protein, it may acquire a new function, right? So for example, all of our antibiotic resistance genes that bacteria evolved that involved like efflux pumps or enzymes or mutant targets to which the antibiotics can no longer bind, those are all initially the result of mutations, right? So mutations aren't always bad. Mutations can increase a population's genetic diversity, and that's absolutely essential for natural selection to act upon. So we have to have that genetic diversity in a population of organisms for biological evolution to occur. So again, you guys, that DNA, the genetic information of all cells, also the genetic information of DNA viruses, and then RNA. There's all different types, you guys, but we just talked about the three involved in protein synthesis, translation. So rRNA, mRNA, and tRNA. RNA is also the genetic information of RNA viruses, like the pandemic coronavirus, like influenza virus, right? And then we'll see in the genetics unit, we'll go through the processes of transcription and translation or protein synthesis. And then next, folks, we move to carbohydrates. Some folks call them sugars, right? We didn't do a lot here. We talked about glucose. And glucose is one of the most common sources of carbon and energy in cells, right? And then we talked about a couple of disaccharides like lactose and sucrose. And then we talked about polysaccharides. We talked about cellulose. We talked about glycogen. And we also talked about modified glucose. For example, the modified glucose N-acetylglucosamine. So polymers of N-acetylglucosamine is called chitin, and we find chitin in fungal cell walls. We find it in the exoskeletons of arthropods. And then we also mentioned the modified glucose N-acetylmuramic acid that can only be made by members of domain bacteria, right? And we said that both N-acetylglucosamine and N-acetylmuramic acid are important components of the backbone of peptidoglycan, the component that provides strength to bacteria cell walls, right? And remember, you guys, only bacteria can make peptidoglycan, and then we'll come back to that in the Unit 3 on cells. Okay, and oh, sorry, I should have showed you these. So here's our N-acetylglucosamine and N-acetylmuramic acid. Alternating NAG and NAM makes up the backbone of peptidoglycan. And again, only bacteria can make peptidoglycan. Lipids, you guys remember, they're not polymers. They're the thing that the property that unites all lipids is they're primarily nonpolar and thus hydrophobic, right? And we talked about 4 groups of fats: so phospholipids, steroids, and waxes, right? And we made a big deal about the phospholipid bilayers being crucial in the fluid mosaic model of cell membranes, right? So this ocean of phospholipids in which membrane proteins are located, right? And we said that the phospholipids are, the individual phospholipids are what we call amphipathic or amphiphilic. They have a hydrophilic head and then these hydrophobic tails here. So when they form the bilayer, they have a hydrophilic side here, hydrophilic side there. But then they have this cool core, kind of this hydrophobic core. And that means that large hydrophilic molecules usually can't diffuse across the phospholipid bilayer. And for that reason, one important function of these membrane proteins is that they're going to act as transport proteins to permit large hydrophilic molecules to move from one side of the membrane to the opposite side. Another lipid, the steroids and sterols. So the steroids have this 4 kind of classic carbon ring structure. A sterol will add a hydroxyl group. So we talked about cholesterol being an important component of cell membranes, for example, of humans. It's a precursor of many of the, for example, sex hormones, precursor of bile salts and vitamin D. And then we said that if we're looking for unique targets of antimicrobial drugs, if we have a fungal pathogen, fungi, instead of having cholesterol, have ergosterol. So that's a cool, unique target of fungal pathogens. So we talked about amphotericin B is a pretty toxic drug that will bind to the fungal ergosterols in cell membrane, cause the membranes to get leaky, and that will kill the fungal cells. And then a second group of antifungal drugs that aren't as toxic are the azoles, like miconazole. Azoles will bind to and inhibit the enzymes that make ergosterol, and you end up with the same thing, leaky cell membrane, so you can kill the fungal pathogens that way. Waxes is the final lipid, you guys. These incredibly long hydrocarbon tails and the fatty acids linked to an alcohol. They're incredibly hydrophobic, and you might remember we said that the mycolic acid, as an example of these waxes, are found in the acid-fast bacteria such as Mycobacterium tuberculosis and Mycobacterium leprae. They have these incredible waxy hydrophobic cell walls. We have to have a special staining procedure, the acid-fast stain, because they don't stain well with a Gram stain. And we want to remember that most of our antibiotics are hydrophilic. So it turns out these acid-fast bacteria, they're often difficult to treat. You have to choose your antibiotics carefully, and often the patient has to be on antibiotic therapy for months, maybe years, right? They're hard to stain, as we said, with a Gram stain, so you have to stain with the acid-fast stain. Because of the waxy cell wall, they're resistant to antiseptics and disinfectants. They're resistant to drying, so they can remain infectious in the environment for long periods of time. They're going to be slow growing, so if you send in, say, a sputum sample and ask the lab to see if the lab can grow Mycobacterium tuberculosis from the sputum sample, you know it's going to be days or weeks before you get culture results and antibiotic sensitivity testing back, right? And we did say, you guys, we did mention that the anti-TB drug isoniazid will inhibit mycolic acid synthesis. So isoniazid is a frontline anti-tuberculosis drug, so you want to remember that one. And then, folks, we move to Unit 3, cells, and you'll recall that we focus almost entirely on bacteria cells because you've either had 2 semesters of A&P or you will have 2 semesters of A&P where you focus for a year on human cells, eukaryotic cells. So we focus mostly on bacteria cells. So just to start out, you guys, we said that prions and viruses aren't cells. So what are some characteristics of cells then? So remember, we said they can synthesize a cytoplasmic membrane or cell membrane or plasma membrane. They're going to have ribosomes for protein synthesis. Cells carry out metabolism, right? Catabolism, anabolism. They'll make ATP. Cells are going to grow and divide, and there's many other characteristics, you guys, but again, we want to stress that viruses and prions are not cells because they violate all of these rules of cells, right? This is a horrible diagram, you guys, and I apologize, but I thought it was probably the best summary for the final exam. Make sure that you know the composition and the function of all the different structures of the bacteria. And again, this is kind of a good concept map. Well, I guess it's not a map, just a diagram here, but this would be a good one to use kind of condensed studying for the final exam. And again, you guys just remember the phospholipid bilayer, fluid mosaic model, phospholipid bilayer, this like an olive oil kind of ocean of phospholipids in which the membrane proteins can move, right? It's fluid, right? It has to be fluid. And remember, you guys, anything that damages the cell membrane usually will kill the cell. And on that topic, we're like, what if we have a bacterial pathogen? What's one way we could kill the bacterial pathogen? Well, one way we could do it is through a process called osmotic lysis, right? So just really quickly, you guys, the bacteria or any organism living in a hypoosmotic or hypotonic environment, that means the concentration of solutes inside the cell is higher than outside the cell, and thus the concentration of water outside the cell is higher than inside. So through osmosis, you get a net flow of water into the cell. The water pressure will build up, and if there's no strategy, then the little cell is going to explode like a water balloon in a process called osmotic lysis. So the strategy many organisms have evolved is to make a strong cell wall outside of the cell membrane, and the purpose of the cell wall is to prevent osmotic lysis, right? And so we made a big deal, you guys, talking about bacteria cell walls, and we said the substance in bacteria cell walls that provides the strength is peptidoglycan. And remember, you guys, peptidoglycan only can be found in members of domain bacteria. And you'll recall, you guys, that peptidoglycan is these alternating chains of NAB-NAB, NAG-NAM, 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 and then they've got these little tetrapeptide tails. And what provides strength in the peptidoglycan is peptide crosslinks between these little peptide tails, right? And we said the bacterial enzyme that forms those essential peptide crosslinks in the peptidoglycan is called bacterial transpeptidase. So this then is going to end up being a unique target for the big group of antibiotics called beta-lactams, right? So you guys will recall that beta-lactam antibiotics like penicillin, amoxicillin, ampicillin, methicillin, and cephalosporins, these are irreversible competitive inhibitors of bacterial transpeptidase. So if you have a patient with a bacterial infection and you give the patient a beta-lactam antibiotic, if the bacterium is sensitive to the beta-lactam binds to the bacterial transpeptidase, stops cross-linking of the peptidoglycan, and then the bacteria will undergo osmotic lysis if they're in a hypoosmotic environment and if they're growing. Because remember, you guys, beta-lactams can't hydrolyze those peptide crosslinks. They just prevent them from forming. And another big gun antibiotic that we really didn't talk much about, you guys, is vancomycin. It too will prevent the cross-linking of peptidoglycan. It has a different mechanism, but the result will be the same. We'll get osmotic lysis. And folks, just remember, Alexander Fleming discovered penicillin. He discovered that a fungus, a mold called penicillium, made this substance that could inhibit the growth of Staph aureus, right? And that was back, I think, like in the late 30s or 40s. Yeah. And then, folks, this is really important, knowing the 3 different types of bacterial cell walls, their composition, and kind of like their medical significance. It's like, why do we care, what kind of cell wall you have, bacterial pathogen, right? So you'll recall, folks, that Hans Christian Gram developed this differential stain, the Gram stain, that lets us distinguish between 2 really important group of bacteria, the Gram-positive bacteria and the Gram-negative bacteria, right? And you'll recall that in the Gram stain, Gram-positive bacteria stain purple, Gram-negative bacteria stain pink, and this is related to significant differences in cell wall structure. So you guys remember the Gram-positive bacteria, in their cell wall they have a thick layer of peptidoglycan, which is very hydrophilic. So hydrophilic antibiotics just diffuse through here, no problem at all. In gram-positive bacteria, we can, in addition, find the lipoteichoic acids or teichoic acids. Those are kind of unique molecules found in gram-positive bacteria. In contrast, you guys, in gram-negative bacteria, you recall they have a thin layer of peptidoglycan. That's really important to remember. And then they have this protective outer membrane that gram-positive bacteria lack. And the outer membrane, you guys, it's important to remember it's a lipid bilayer. The inner leaflet here is made out of phospholipids. The outer leaflet here, you guys, that's where you find a toxic lipopolysaccharide, LPS, right? We've called that endotoxin because we know it can trigger a horrible cascade of events that can lead to sepsis and septic shock and death, right? In this outer membrane, right? Let's see my cursor. Okay, we're going to have protein porins, right? So if you have a hydrophilic antibiotic, for it to be able to cross through the outer membrane, it has to be able to cross through those outer membrane porins. And you'll recall we've said in some gram-negative bacteria, the porins won't let some antibiotics across. So for example, in most gram-negative bacteria, penicillin can't cross through the outer membrane porins. So we said in general, most gram-negative bacteria are naturally resistant to penicillin, right? But we said that there are semi-synthetic beta-lactams like ampicillin and amoxicillin that have been chemically modified so that they can cross through the outer membrane porins, right? Okay, let me see here. So yeah, do you remember, you guys, in the gram-negative bacteria, they're the ones that have the toxic lipopolysaccharide. And then we have our acid-fast bacteria with that outer with that waxy cell wall, the most resistant bacterial cell wall of all, right? And do you remember mycolic acid is one of the waxes, and isoniazid will help inhibit the mycolic acid, one of the frontline anti-TB drugs. And this was just from your handouts, you guys, so gram-positive. This is the cell membrane here, just for reference. So gram-positive, thick layer peptidoglycan with lipoteichoic acids, teichoic acids. Gram-negative bacteria, thin layer peptidoglycan, outer membrane, lipopolysaccharide, toxic porins here. And then the acid-fast bacteria with this really thick, waxy cell wall. And again, folks, we should always go, why do we care? Well, we want to remember that the Gram stain is like step 1 in identifying an unknown bacterium, so it's crucial in identification of an unknown pathogen. And remember, you guys, knowing the type of cell wall is going to be absolutely crucial in choosing your antibiotic, right? So we want to remember, for example, like penicillin can't cross the cell walls of most gram-negatives. Many antibiotics can't cross the cell wall of acid-fast bacteria, right? So we have to know the type of cell wall to choose our antibiotics correctly. And this was just a reminder, you guys, of the lipopolysaccharide, the endotoxin, which causes the massive cytokine release from our white blood cells, that cytokine storm, which leads to vasodilation, leaky vessels, hypotension, decreased blood flow to tissues and organs. We get a triggering of DIC, disseminated intravascular coagulation. We start forming clots everywhere in our vessels. We use up our clotting factors and we start to hemorrhage. So we can rapidly progress to shock and then death, right, if we have a massive release of endotoxin from gram-negative bacteria growing somewhere they shouldn't be growing. We're kind of moving our way outward from the cell wall. Now we're going to look at the glycocalyx, right, the sugar cup. And remember, you guys, there's 2 kinds of glycocalyces, the slime layer and the capsule. The capsule is antiphagocytic. The slime layer, you guys, helps Microbes stick to a surface in their environment, and they play a really important role in forming biofilms. And the one that we actually gram stained in lab was our tooth biofilm, right? Formed, for example, by Streptococcus mutans. And we said the biofilm, it permits the bacteria to stick to our teeth so we don't swallow them. And then once they're stuck to our teeth, especially if we eat a lot of sugar, they start fermenting it. They make lactic acid. The acid helps decalcify our teeth, can increase our risk for dental caries. And we know if these tooth biofilms, this plaque, isn't cleaned regularly from our teeth, the metabolic waste products can cause chronic inflammation of our gums, and that can lead to periodontal disease. And there's also some evidence suggesting that poor oral health, chronic inflammation of your gums, might also contribute to cardiovascular disease, because you get these oral bacteria being constantly shed into the bloodstream, and they may indeed cause inflammation of blood vessels. And remember, you guys, this concept that bacteria living in these thick biofilms are much harder to kill than free-living or planktonic bacteria. They're often in the stationary phase of growth, so they're naturally more resistant to antibiotics. The layers of the biofilm provide protection against phagocytic cells, and the layers of the biofilm can also create a diffusion barrier against antibiotics and against antibodies or immunoglobulins. So more structures on the outside of the cell, you guys. Some bacteria have these protein hair-like filaments that protrude outward called fimbriae, and at the tip of the fimbriae there will be special proteins called adhesins, and the purpose of the adhesin is to bind to specific host cell surface receptors. So if we have an invading bacterial pathogen, they need to stick to our cells, right? And they would do that through the adhesins binding to our host cell surface receptors. And we said we can block this attachment if our body makes antibodies, and here you guys are just little V's. The antibodies can bind to the adhesins. That's going to block attachment. And remember, you guys, we said that antibodies that block attachment are called neutralizing antibodies, and they're powerful because it can prevent infection. We want to remember, you guys, that bacteria, because they are prokaryotes, are going to have 70S ribosomes, right? And we know that then is a great target for our antibiotics. So you recall that we talked about 4 groups of antibiotics that target and inhibit the bacterial 70S ribosome. And those antibiotics included tetracycline, the macrolides like erythromycin, azithromycin, chloramphenicol, and aminoglycosides. And you guys, on the final exam, I love antibiotics, right? So make sure you're feeling confident with the antibiotics and their targets and which functions they would inhibit. And this is important, you guys. So why doesn't tetracycline kill us, right? Oh, and that's so totally misspelled. Oh my gosh, you guys, isn't that embarrassing? Okay, tetracycline. Ay, ay, ay. Okay. Let's see if I can get that changed here. Okay, without making a mess of this. Okay. So do remember, you guys, that as eukaryotes, we have 80S cytoplasmic ribosomes, right? It is true through the endosymbiotic theory, we know that our mitochondria have the 70S-like bacterial ribosomes, but at safe therapeutic levels, tetracycline, for example, it won't shut down our 80S ribosomes, and it doesn't get in high enough concentration in our mitochondria to shut down the mitochondrial 70S ribosomes. So it's really important, you guys, that the eukaryotic cytoplasmic ribosomes are 80S. The prokaryotic bacterial cytoplasmic ribosomes are 70S. So that's a great target for our antibiotics. Luckily, you guys, only a few groups of bacteria can make endospores. And remember, you guys, the endospores are these really tough resistant structures that are formed to protect a copy of the chromosomal DNA when environmental conditions deteriorate. So members of the genus Bacillus can make endospores, such as Bacillus anthracis, which causes anthrax. And then all members of the genus Clostridium can make endospores. So we talked about Clostridium tetani causes tetanus, Clostridium botulinum causes botulism, Clostridium difficile C. diff that causes a pseudomembranous colitis, and can actually cause perforation of the intestine. So you recall, folks, the process of forming the endospores is called sporulation. So here's Clostridium tetani, and here's the endospore right here. That's sporulation. And then usually the metabolically active cell dies and releases the endospores into the environment. Often they're found in the soil, the dirt, and there they can last for decades, centuries, maybe thousands of years, right? And then if the endospores then are introduced into a more beneficial environment, let's say where it's moist and warm and nutrients are rich, then they can germinate, and then that's going to release the metabolically active bacterium. And if that germination occurs in our tissues, right, often these endospore formers, they often can make powerful exotoxins, right, that can cause great harm. So this little poor little baby, you guys, this poor little baby has neonatal tetanus caused by Clostridium tetani. Neonatal tetanus usually occurs when the umbilical stump, which is going to be anaerobic, gets contaminated with endospores. And often it's just from just plain dirt, right? The instruments used to cut the umbilical cord literally are dirty. Endosymbiotic theory, again, you guys, we already stated this. So evidence supporting the endosymbiotic theory that mitochondria and chloroplasts evolved from bacteria is they have 70S-like ribosomes, they have circular DNA that looks like bacterial chromosome, they're self-replicating, the membrane arrangement is consistent, and they have about the same size as bacteria. This was phagocytosis. This was actually part of our discussion of the how bacteria could become endosymbionts, but for non-specific interior defenses, you guys, you do want to review phagocytosis. Okay. And again, this was just looking at some of the eukaryotic cell slides, you guys. We said that eukaryotic cells, they might be motile, like with our protozoa, they can have pseudopods, they can have flagella, they can have cilia. And then again, folks, just kind of, this is actually kind of leading into the non-specific interior host defenses. We want to remember the beautiful ciliated epithelial cells found in the upper respiratory tract and their important role in the mucociliary escalator to get rid of microbes trapped in that mucus blanket, and also the important role of ciliated cells in the oviduct fallopian tubes of women. And remember, you guys, we said that anything that damages these ciliated cells, like in the trachea, if we have a viral infection, say with influenza or the coronavirus or from smoking, that will damage the mucociliary escalator, and so that means you're going to be at higher risk for secondary bacterial pneumonia. And we said in women, if they have silent or untreated STIs, like with Neisseria gonorrhea or chlamydia, those bacteria can cause chronic inflammation in the oviducts and damage ciliated cells. So that when an ovum is ovulated, right, the ovum gets trapped in the damaged oviducts. And if the woman is inseminated, the sperm, which are motile via flagella, can still move to that kind of like that stuck egg and fertilize it. So the problem is then you can end up with an ectopic pregnancy, which is life-threatening. The woman can end up with pelvic inflammatory disease. She can end up with fertility later in life, right? So we need to protect those beautiful ciliated cells. And then the last couple of slides, you guys, in the Unit 3 was on transport of substances across membranes. So we can have diffusion of small nonpolar substances directly across the phospholipid bilayer, but if we have large hydrophilic substances, they're going to have a membrane protein, right? So you guys will recall we talked about passive transport, and passive transport doesn't require an extra input of energy because the substance being transported is moving down its concentration gradient from high to low. Okay, if it's a large hydrophilic substance, we'll have what's called facilitated diffusion, where there's a membrane protein that's helping the substances move across the membrane. And also, this always made me feel better, you guys. Aquaporins are protein channels through which water can diffuse. And then the last type of transport, you guys, and we just kept it really simple, active transport, we said this is when the substance is moved across the membrane against its concentration gradient. So you're moving the substance from an area of low concentration to high. We said this always requires a membrane protein, and it always requires an input of energy. So it could be that ATP gets spent. It could be that maybe like a proton gradient is going to be used as a source of energy for active transport. So you guys, I think that's it. So I know that was really quick. And again, you guys, remember this is nowhere near exhaustive, but hopefully just kind of maybe just triggering some memories. Do make sure, you guys, that you use your lecture study guides. That's the best way to prepare for the final exam. And then, folks, tomorrow, Saturday, I'll try to get some audios made, at least on the second PowerPoint, and Sunday I'll try to get an audio made for the third PowerPoint. Okay, you guys take good care.